This chapter explains how biopsychologists conduct their research. Although this chapter has two major parts, one that deals with methods of studying nervous system and the other that deals with methods of studying behavior, we will look at the first part only. In this module, we will look at 5 different methods that are used to visualize the living human brain. In subsequent modules, we will look at more invasive procedures that are done with also living human brain, but in some cases posthumously. Prior to the 1970s, biopsychological research was impeded by the inability to obtain images of the organ of primary interest, which is the human brain. Conventional X-ray photography is pretty much useless for this purpose, because when an X-ray photograph is taken, an X-ray beam which passes through an object and then onto the photographic plate, each of the molecules through which the beam passes absorbs some of the radiation. Therefore, it is only the unabsorbed portions of the beam that reach the photographic plate. That's why X-ray photography is only effective at characterizing internal structures that are different from each other. Basically, if you go through an airport checkpoint, they would not be able to look at different cloth underneath your sweater because it's the same substance or same structure. But if you have some other piece of plastic or metal, then that is something that the X-ray photography would be able to spot. One of the ways around this problem is cerebral angiography, which is a form of contrast X-ray technique, which requires an injection into one compartment of the substance that absorbs X-rays less than or more than surrounding tissue. This substance then heightens the contrast between the compartment and the surrounding tissue. And this allows the infusion of radio-opaque dye into cerebral artery so that we can visualize the cerebral circulatory system during the X-ray photography. And here you have a picture, a very nice picture of what cerebral angiography looks like. Again, cerebral angiography is a contrast X-ray technique that visualizes the cerebral circulatory system. And here you can see the vascular system all throughout. This is typically used for when individuals want to locate tumors around the brain. And if we see that there's a displacement of the blood vessel from the normal position, then it could be an indication of a tumor pressing against it. You can see how beautifully the arteries basically flood through here. If there were to be a tumor that's pressing against this, then we would not see an artery in its typical fashion. And this could bring about effects depending on the part of the brain that's being deprived of oxygen. CAT scan, X-ray computed tomography, basically revolutionized the study of the living human brain in the early 1970s. During cerebral computed tomography, the neurological patient lies with his or her head positioned in the center of a large cylinder you can see here. On one side of the cylinder is an X-ray tube that projects an X-ray beam through the head to an X-ray detector mounted on the other side. The X-ray tube and detector automatically rotate around the head of the patient at one level of the brain, taking many individual X-ray photographs as they rotate. The information in each X-ray photograph is combined by a computer to generate a CT scan of one horizontal section of the brain. Then, the X-ray tube and detector are moved along the axis of the patient's body to another level of the brain, and the process is repeated. The scans of 8 or 9 horizontal brain sections are typically obtained from a patient, and combined they provide a 3D representation of the brain. Here you can see the horizontal CAT scans, which combined in a computer would render a very detailed three-dimensional representation of the brain. The success of the CT scan was a very good platform for the development of other techniques so that we could obtain better images of the inside of a living body. The MRI is clearer than CAT scans. It provides a high-resolution, two-dimensional and three-dimensional images, and it is very good at providing relatively high spatial resolution. That is, the ability to detect and represent differences in spatial location. Because of this spatial resolution provided by the MRI, this is often used to locate growing tumors. As you can see in this picture here, our ability to spot the tumor here would allow neurosurgeons to measure how deep it is and would use language such as medial to the hippocampus or lateral to the third ventricle. And based on that, the MRI assists for preparation. If too close to a part that would control vital functions, then this MRI would help the neurosurgeon make a better informed decision as to whether to operate or not. The three previous brain scanning techniques were considered structural brain imaging techniques. Structural meaning that it would just be a static or photograph form of what our brain looks like. The PET scan was the first brain imaging technique to provide images of the brain activity, and these type of brain imaging techniques are called functional brain techniques. These allow us to look at the brain as it is stimulated and as it is responding to stimuli. As you can see from this photograph, PET scans are not really images of the brain. Each PET scan is merely a colored map of the amount of radioactivity in each of the tiny cubic molecules that compose the scan. One common version of the PET scan is what we call the radioactive 2DG, which is deoxyglucose. This is injected into the patient's carotid artery. That's an artery of the neck that feeds the ipsilateral cerebral hemisphere. Because of its similarity to glucose, which cells consume at high rate, are rapidly taken up by these active cells. And the reason why is because they are consuming energy. However, unlike glucose, which is metabolized, radioactive 2DG cannot be metabolized. And because of that, it accumulates in these active neurons or in associated astrocytes until it is gradually broken down. Each PET scan in here has an image of the levels of radioactivity indicated by color coding. Red and yellow represents activity. Here you can see pictures of the occipital lobe at the bottom of this in here. Whereas on the left, this would have been an individual that is being exposed to something with their eyes open. Perhaps this individual over here has his eyes closed and they're not having a lot of activity. Another form of functional brain scanning technique is what we call the FMRI. This is arguably the most influential tool of cognitive neuroscience, and it is widely used for medical diagnosis as well. FMRI produces images representing the increase in oxygen flow in the blood to active areas of the brain. FMRI is possible because of two attributes of oxygenated blood. First, active areas of the brain take up more oxygenated blood than they need for their energy requirements, and thus oxygenated blood accumulates in active areas of the brain. Second, oxygenated blood has magnetic properties that influence the radio frequency waves emitted by hydrogen atoms in an MRI. The signal recorded by fMRI is called the BOLD signal, the blood oxygen level dependent signal. fMRI is superior to a PET scan, and four of the advantages are that it has nothing to do with being injected into the volunteer. 2, it provides both structural and functional information in the same image. 3, its spatial resolution is better. And 4, it can be used to produce 3D images of activity over the entire brain. Unfortunately, it does have its drawbacks, and one of them is that it has a poor temporal resolution. That is, that it is poor at specifying the timing of neural events. Indeed, it takes 2 to 3 seconds to create an FMRI image, and many neural responses, such as action potentials, occur in milliseconds. You can see how this is a problem when looking at a functional image. As amazing as PET scan and fMRIs are, and have allowed cognitive neuroscientists to create images of the activity of the human brain, these have a weakness, and that is that they can be used to show a correlation between brain activity and cognitive activity, but they cannot prove that the brain activity caused the cognitive activity. For example, we can demonstrate that a specific part of the brain becomes active when volunteers are exposed to disturbing photographs, but it cannot prove that that specific part of the brain causes an emotional experience. There may be many other explanations. One way that we can support the hypothesis that it is that specific part of the brain that is an area for emotional experience would be to assess emotional experience in people lacking that functional part of the brain. For example, by studying patients with damage to that specific part of the brain, or studying healthy patients with that specific part of the brain that has somehow been turned off. And that's exactly what TMS does. It allows us to turn off areas of the cortex or also turn them on. Please note that this is not a measure of neural activity. It provides an experimental probe to alter neural activity, as the transcranial magnetic stimulation applies a brief but strong magnetic field that alters neural activity. In fact, we've noticed how individuals claim that safely they could stimulate parts of the brain that are known to control speech, for example. And they are asked to count letters, to tell the letters or count numbers 1 to 10, and while speaking, they're stimulated by this magnetic force, and we are able to impair their speech. We can also get them to move their arms involuntarily by simply stimulating with a magnetic force the part of the brain that would control that part of their body. So these, we can either activate or deactivate brain structures and simply observe changes in behavior, therefore allowing us to have strong evidence for causal effect of specific part of the brain on behavior.